Hello, people. Happy Friday. Hi, guys. Happy Friday.
Hi, Mr. Long.
I think I missed some people popping in, so let me see who I'm missing here. And yell if you are here. Yell if you're here. Amra Bati. I don't see you on the list. Okay. Elena Her. Are you in? Elena Her? Nope. Okay. Claire. Olivia Kraby, not here. And then, oh, that's it. So who just came? Oh, Julia, she was already in. So once again, I got Amra, Elena Her, Claire Hovorka, absent. I don't hear anybody shouting, so I'll assume that's the case. Okay, everybody, camera on, please. Keep them on. I'm just doing my attendance quickly. Get ready to write stuff. Get yourself all prepared to draw pictures and writing and all of that stuff. And I just remembered something I forgot. Perfect time to remember it right when class starts. Elena is excused. Let's see. And Amra. Okay. We did not quite finish our respiration chapter, but we're almost done. So we're going to wrap that up and then we'll dive into photosynthesis. I have posted the beginnings of a respiration lab. At least this is as far as I've gotten on it. I spent about 3 hours so far on this thing. And I'm sure you will show your appreciation for all of my work prepping this thing, that you will do a super great job on it and everybody's going to turn it in on time and all of this, much better than the last one. Yay. Because we love pivot labs. They're super fun and they allow us to do science virtually. Right? Right, Anna Hoagland? Right. No faults, she lies. Enough of your mouth lies. Okay, who just popped in? Amra? Okay, I gotta mark her present. I'll do that later. Okay. All right, I just need to pull up the PowerPoint here. I kind of forgot we still had to do that respiration thing, so I don't have it ready. Okay, here it is. We were talking about other compounds that we did this at the very last minute, like right when class was over, and people were probably already shutting down. So I'm just going to mention it again real quickly. We talked about how different molecules feed into different biological molecules feed into the respiration pathways. Because everything we always talk about is glucose. We always talk about glucose all the time. And it gives people this misconception that everything is glucose. That everything, all the ATP we get is from glucose. So, we talk about how many ATPs per glucose and all of this stuff. So glucose is clearly king, but other molecules are in our diet too. So they're also important. Especially if somebody's on like a keto diet or Atkins diet where they're hardly eating any carbohydrates. So then all of a sudden now fats and proteins are super important. They're the dominant source of energy for those kinds of people. All right. Okay, so we said that proteins, the amino acids have different sizes and they get broken down. The amine gets stripped off and then they get broken into various size carbon molecules, which then feed in different places. So if it's a 3 carbon molecule, oops, I can't draw here. If it's a 3 carbon molecule, oopsie, why does this look different than normal? Shouldn't I have a pen option here? Huh. Okay, it works now. Okay, so if it's a three carbon molecule, it'll feed in at pyruvate. If it's a 2 carbon molecule, it'll feed in at acetyl-CoA, because pyruvate is 3 carbons. So it's an easy transformation to transform it over into pyruvate. Acetyl-CoA is 2 carbons, so it's easy to transform the 2 carbons of, let's say, glycine into acetyl-CoA. And then various size molecules that have, the ones that have larger R groups will feed into the larger molecules in the citric acid cycle, whether it's 5 or 4, most likely 4 carbons. So the amine gets broken off and it gets eliminated because that's a toxic compound. Ammonia is very, very dangerous and toxic and poisonous. And that gets, how do we get rid of the nitrogen, by the way? I talked about that last time. I don't know if people are still listening. How do we get rid of this nitrogen? Say it louder.
Urine.
Urine is correct, yes. I got to turn my speaker up here so I can hear you guys better. Okay, thank you for that. Fats, same thing basically, except there's no nitrogen in fats. The glycerol gets split apart from the 3 fatty acids in the triglycerides, because most fat in the food is triglycerides. I mean, that's really just about all of it. I mean, yeah, we eat phospholipids too and stuff like that. Yes, we eat cholesterol, but we're talking about triglycerides here. So the glycerol clearly feeds in here at a 3 carbon stage. The easiest and most obvious place is glyceraldehyde 3-phosphate, because that's basically like a glycerol and a phosphate stuck on it. So it's very similar to glycerol already, so you can feed it in there. Super easy. Fatty acids are like, super long chains of carbons. They could be like 18 or 20 carbons or whatever, or they could be shorter, like 8. They get chopped up into chunks of 2. That's called beta oxidation. So you chop them into chunks of 2 carbons each, and those feed into acetyl-CoA, which is 2 carbons. All right, so that was the last thing we talked about. Any questions on that? I would say this is not something necessarily to memorize. Like, I wouldn't memorize this whole big spiel here. I just would be real sure I understand the concept. The big picture concept is that we have different sources of food molecules. We have carbohydrates like glucose. Starch is essentially glucose. We have protein, we have fat. All of those things contribute for us to make ATP. And here's how they do it. So I wouldn't necessarily memorize it. I would more like, understand that these feed in at various different places. So if you don't remember that, let's say glycerol feeds in at glyceraldehyde 3-phosphate, I don't care about that. I just need you to understand the big picture. Doing all right on that? Okay, let's move on. Am I recording? I think I'm recording. Okay. So this I don't think I talked about yet. So I'm going to go through this a little bit more carefully, although it really won't take that long, because it's actually pretty easy to understand. It just looks complicated. It looks worse than it is. This is not so bad. Basically, what I'm saying here is what we're trying to show here is that, oopsie. What we're trying to show is that we can regulate this whole process. We can speed it up. We can slow it down. We can control it, which is good, because if we have a lot of ATP getting produced in a cell, we might want to throttle this down a little bit, because you don't want to make too much ATP. First of all, you cannot store ATP. You can't like save it for later. Have you ever noticed like you can't go to like Walmart or Sendix or whatever and buy an ATP energy bar? There's no such thing. You can buy carbohydrate bar, you can buy protein bar, you can buy something with fat in it, but you can't, or a mixture, but you can't buy like ATP straight up. And the reason why is because ATP is really unstable. It's not something we can eat. It would break down and it would fall apart before it would ever get into the bloodstream. And it would probably be dangerous because it's a high energy molecule. So our cells have to make the ATP that they need at the time they need it. But if you already have a lot, you want to slow down the making of the ATP. So the way that's regulated is a number of different options. So first of all, ATP itself can can act with the process that we call negative feedback. So negative feedback is if you've made enough of a product, the product itself will shut down the production of itself. We have enough ATP, so the ATP then turns off the making of more ATP because we have enough. The way it does that is it feeds in at phosphofructokinase. I told you phosphofructokinase is the one most important enzyme in glycolysis that I'd like you to know. I asked you to memorize all of that stuff in glycolysis, but like for the AP test in May, that's the one I would remember out of all of them. Even if you forget all the rest, that's okay. That's not a problem. But phosphofructokinase is super important because we can regulate the whole process, the whole spiel at that level. If ATP binds phosphofructokinase, it shuts it off. It has a negative effect here. So this is negative feedback. It inhibits it, which then shuts this down, which will shut this down, which will shut this down, and then ATP will go down. And as ATP is reduced, this goes away, which means this negative feedback will stop, and this will get turned on again. It's a beautiful system. As you build up more ATP, you slow down phosphofructokinase, which slows everything down then and stops everything. And as the ATP disappears, you start it up again. Now, I don't want you to think that it's like this all or none thing, that it's like on or off, or like we use up all the ATP and it's all gone and then we decide to make more. No. We always keep that ATP very, very constant. That's part of that homeostasis business in living organisms, is to keep that ATP at a good level, that the cells always have enough, that, as much as they need. Because if you remove ATP from a cell, if you withhold it, the cells will die. And especially in our body, the brain cells will be the first to die. So that's a system of negative feedback where ATP can shut down phosphofructokinase and then essentially shut down this whole process, will get stopped to regulate itself. Negative feedback. Is that clear? I mean, I kind of talked pretty fast, but I thought I said it pretty well. Like, I don't know. I was listening to myself at the time and I was thinking to myself, my Mr. Volk, you're really eloquent the way you're describing that. That's what was going through my brain. Was that going through your brain too? Yeah. I don't see anybody really responding here. This is super fun. Super fun. It says Claire is joining. I hope she's going to get in. Okay. Nope, didn't make it. All right. Any questions on this or comments? Okay, the other way we can regulate it is that citrate. If citrate builds up, because if there's too much citrate and like the Krebs cycle is not breaking down the citrate fast enough, that'll like throttle down phosphofructokinase too. So citrate can inhibit PFK also. It is another way that a feedback can happen. Citrate is the reason we call it the citric acid cycle, instead of the Krebs cycle sometimes. So. And then a third way that we can talk about is that AMP, adenosine monophosphate, versus ATP can stimulate positive effect on phosphofructokinase, can turn it on. Because if there's a lot of AMP, that means there's a low amount of ATP. The less ATP there is, the more AMP there is, and the more, and the other way around. The more ATP there is, the less AMP there is. You might say, don't you mean ADP? Because when ATP is used up, it forms ADP, which is adenosine diphosphate. Yeah, that would make more sense. Unfortunately, reality is that AMP controls it here, and it's much more difficult to explain why AMP has that relationship with ATP. However, I knew you would ask, and I'm not going to go over it because you don't need to know it for me or for my test, but here's the answer. So I gave it to you here just in case you're like dying to know. What does AMP have to do with ATP? Well, this explains it. Because I was afraid somebody would ask this and then I wouldn't have the answer for you. Well, there it is, bam. And that's the end of that chapter. Any comments, questions, concerns, praise for the teacher, reprimands for the teacher, anything? Oh, come on. Okay, before we start the next thing, which is photosynthesis, which is going to be super fun because everybody loves photosynthesis. can we all have the cameras on for a minute and maybe like open up your mic if you're going to say something? I just want to take like a minute to ask you guys some questions. Ryan, you there?
What do you need, Mr. Volk?
I just wanted to see your lovely face. Anyway, thank you. Thank you. Here's my first question, and maybe it's the only question that I have. Are you guys okay?
That's a very good question.
Are you okay?
Good question.
Because the feeling I'm getting, my gut feeling right now is that no, you're not. That you're not okay. That's what I'm seeing from the way you guys look at the screen, the way you guys talk, the way you turn stuff in or don't turn stuff in. I'm a little bit worried. So is everybody struggling and down and lethargic and all that? Or is anybody actually like feeling like virtual learning is working? Like, are you guys all like frustrated with the virtual thing and it's too much and it's Rona sucks? Well, yeah, I mean, I would agree. Oh my God, the chat was already going. I wasn't looking at it. I'm sorry. I'm sorry that you're in pain and I'm sorry that you're dying. If you could do me a favor though, could you please not die during my class? You know why, right? You know why.
It's too much paperwork.
Because of the paperwork, I know. It's Friday. I mean, I want to go home and I want to get a fish fry. And if kids are dying again in my class, I mean, for the first time, I meant, yeah. Okay, that hit hard. Not okay. Okay with what? I mean, like with life and school and like just the state of the world. I mean, I know COVID sucks and everything. Like, you know, somebody said Rona sucks. Well, no kidding. I mean, everybody hates it. But, you know, I don't know if you guys watch the news. I've not been watching the news a lot because it makes me depressed. So I've been like purposely just listening to music in my car, and I don't, at home, I barely look at the news. But I've been hearing a lot of stuff about the vaccine. The vaccines are coming out and they're going to be available soon. So that's really encouraging to me. Like that makes me feel really good about the state of the world. No, I mean like your mental health. I meant your mental health is what I'm asking. It's not working, oh boy, that's what I thought. See here, I thought I kind of felt like I was actually doing an all right job with this. Like I felt kind of good about myself, but now, now, maybe not so much. Haven't had motivation, virtual is that bad, life's just boring. See, I think, yeah, I'm sorry, guys. Okay, I'm having trouble. Okay, so somebody said, Volker, are you okay? Well, I wasn't in August and in September, and even like all the way at the beginning of November, I was not okay. Like I had like serious issues with mental health. And I mean, I'll tell you something. In August, I thought I was going to die. Like I didn't think I was going to make it, like not physically, but like up here. But now I'm taking my pills, and apparently the titer is up to where it needs to be. And I'm actually kind of feeling guilty that I'm doing pretty good. Like I'm feeling, for the last month, I felt pretty good. And I go home and I watch my office shows and I eat my dinner and I'm content. My wife is nice to me. You know, I mean, my dog, I got a good life. So I'm feeling kind of guilty that here I take it for granted that you guys are okay too. But then I find out like, oh crap, you guys are not. And now I feel bad because here I am, I'm coming to school, I'm having a blast. I'm talking about phosphofructokinase, which is like super fun for me. And then I realize, oh, maybe it's not as much fun for them. So, I am wondering and I'm hoping that maybe we come back soon. I mean, they're talking about stuff like when we come back, and I don't know if we're coming back after break. Nothing's been decided, like nobody knows yet or whatever, but at some point we're going to be back, and I think that's going to help. I think that's going to be good. I don't know. I'm going to have to read this chat later because it's too much, and I'm sorry if I'm missing, like, if somebody's like telling me like really important stuff about their mental health and I'm supposed to act on it. Like if I missed it, I'm sorry. It's just way too many things to read right now. And now you're going to make me cry, Leilani. Jeez. All right. Anyway, I know what'll cure this. I know it'll cure this. Plastoquinone. That always cheers me up. Let me tell you something. Plastoquinone is, no, Anna, really? Did you really? You're not going to be in my class anymore?
No, I don't think so.
You're the one who you're like the one person that's willing to tell me when I'm stupid. You're like the one person that actually is honest with me. I need that.
I'll still email you.
You're the reason I get my grading done on time. You're the reason I get stuff, typed in different campus, because you always yell at me for not doing that. What's going to happen when you're gone? Like nothing will ever get done anymore. I'm sorry. Okay. Anyway. All right. Photosynthesis. Thanks for sharing, guys. Thanks for typing stuff. I'll read that stuff later because it saves the chat. But and I'm sorry if I missed something important. I don't know how to get out of this. It won't let me escape. There it goes. All right. Did I post this on? Oh my God, I might not have posted this. I got to check Google Classroom. I'm sorry. I'm an idiot. Okay, here we go. Oh, that's big. Okay, 7th hour.
I think you did post it with.
I posted photosynthesis. Let's see.
Yeah, it's like important stuff for photosynthesis.
Oh, I did. Thank God. Okay, I totally forgot that I posted that. It's all in one. It's all in one thing here. So the notes are there. Flow chart, chapter 10 PowerPoint. That's the thing I'm going to be working on now. So I'll go through that. Light reactions, dark reactions. Those guys are there. So everything's here. Okay, so you guys can. And reading guide I said was optional. Did I say that? You could still like look through it and see if you know the stuff. You don't have to write it all out or whatever. So anyway. Oh, and I posted a respiration lab for you guys to work on this weekend. So you're going to be doing that at home. Well, you do everything at home. I was saying that. I'm always like, oh, when you get home, you have to. Okay, yeah, you're already sitting at home. Too soon. So respiration lab. I'll talk about this at the end of the hour. So if it's like 10 minutes left of class and I'm still going on about borophyll or who knows what, can you guys remind me to talk about this? Because we will be doing a lot of this outside of class. Because like I said, on Tuesday I want to finish photosynthesis, and on Friday I want to give you a test. So you guys remembering that 1 week from today, test on 2 chapters, respiration, photosynthesis.
Can we not have a test that day? Because that's like my birthday. So can we not?
You're a traitor. You're leaving us. And now we're supposed to do you favors?
I'm not dropping the class. I just am staying home.
You've left me. We're already at home.
I don't want to come back. I don't want to be around people.
Yeah, but me, everybody wants to be around me. Come on. All right. So we'll talk about that at the end. Okay. Photosynthesis. Bam. No, like all full, like the what's it called? The full virtual or whatever. That means you're like taking those other classes, right? You're not in my class then, right? Or are you still in my class?
No, I'm not going to be in your class anymore.
Yeah, see, that's what I meant. All right. All right, so some people say photosynthesis like respiration backwards. So, if we got respiration, we're going to take glucose. There we go again with the glucose. And we make CO2 and water. And by the way, like, we know all the parts now. Like, don't do silly stuff in chat. Now you guys got to focus because we're going to go through this fast, okay? We're going to go through this pretty quickly because a lot of this is review. So I need you not to be in the chat acting silly now. I need you to be like really have the brain turned on and be in the zone here. Okay, so the glucose thing we can talk about, but everything like glucose is processed where? Like we know the process. Is that Krebs cycle? Is that electron transport chain? Is that glycolysis? You know, where's that? So we know this is the part where you shouted right away. We know it's glycolysis. Thank you for shouting it silently. Okay. Oxygen. We know where oxygen comes into play. Where does oxygen come into play? Is it glycolysis? Is it the Krebs cycle? Is it oxidative phosphorylation? Where is it? What?
Electron transport chain.
Thank you. That's exactly right. The electron transport chain. So at the end of the electron transport chain, oxygen is the final electron acceptor. So we know this part. We understand all the parts now. CO2. Where does that come into play? Where is CO2 produced? Is it glycolysis? Is it Krebs cycle? Is it electron transport chain? Where do you get the CO2? We know this.
Krebs cycle.
Krebs cycle. Whoever's shouting the answers, I can't tell. Is that Leilani? Thank you. I should acknowledge that. So sometimes I can't see who it was. Thank you, Leilani. Okay, so that's Krebs cycle. Okay, water. That one's a little bit more tricky. Where's the water made? You remember that one?
Electric transport chain.
Yes. Where the oxygen comes in, that oxygen goes into water. Those O's from the oxygen go into water molecules. So that's where that's made. So we know all the parts of respiration. Okay, and then photosynthesis, we draw it out like this. We say, well, CO2 plus H2O makes glucose plus O2. It's like, oh, it's this exact thing backwards. You know, well, it's not. It's not. There are similarities, but the reactions and the molecules and the enzymes and the structures in the cell are very, very different. There are some similarities, like there's still ATP synthase. There's still an NADH-like molecule, except now it's NADPH. And it's easy to remember because P, like photosynthesis, starts with P, NADPH, okay? Whereas NADH is in respiration, NADPH is in photosynthesis. And the other thing that people always confuse or mess up and forget about is that plant cells, obviously plant cells have chloroplasts where they do photosynthesis, but they also have mitochondria. They do the whole respiration thing also, just like animal cells do. Same kind of mitochondria, they do same processes. When, in the dark at night, plants respire. They actually use oxygen, just like we do. But when it's light out and the sun's shining down on the leaves, then they produce way, way, way more oxygen than they consume. So during the day, yes, the net result is an output of oxygen, but they still, they still have those mitochondria. They're still doing all their respiration. So in our lab, we're going to be looking at plant cells respiring. So we're going to look at germinating peas. And those germinating peas are not grown enough to actually do photosynthesis. They don't have leaves yet. They don't have really a lot of chlorophyll yet. They're just doing respiration. So we're treating them like almost like an animal would just take in oxygen instead of producing it. All right. So in terms of leaf structure, we should review this a little bit, and like cell structure. In the leaf, if you look at the cross section of a leaf, there's like the outer epidermis and stuff like that. We're not going to worry about all that anatomy, but I will point out a few things. This is called palisade mesophyll. Palisade mesophyll spelled correctly by the closed caption. There you go. This PowerPoint is really smart. This part here is called spongy mesophyll. And the mesophyll cells are just the cells that have like most of the chloroplasts, and that's where most photosynthesis takes place, in these two layers. Spongy just means that it has spaces for air to get in there because there's these stomata, which are like little openings. They're like doors where if the cells either take on water or release water, they can open and close, kind of like this. And when they open, air can come in, and the air brings CO2 with it, which is what the plant cells need for photosynthesis, for the Calvin cycle. So then these spaced out cells with the air and the CO2 between them have access to CO2 here that can then diffuse into the cells and be available for the Calvin cycle. And they produce oxygen, and the oxygen then leaves through the stomata And those stomata, like I said, can open and close. So if it's really dry and there's a lot of water loss through the stomata, they can close up and like photosynthesis will slow down a little bit. Or if they're open, you know, on a day when there's plenty of water, if there's rain or whatever, they'll open up and let all the gas to get exchanged. So that's the cross section of a leaf, like this way on the leaf. If you cut it and look at it from the side, that's what that would look like. And if we look at like one cell, or if we look at one chloroplast, let's look at one chloroplast. That's these little green things here, and we blow that up. You know, obviously the plant cell would have lots of chloroplasts. You know, if you look at the plant cell, we don't have a picture of that here. We have the vacuole in the middle. We know all this. And then the chloroplasts are, you know, all over the place around that vacuole. And they're doing cytoplasmic streaming. So, you know, those little microfilaments are moving them around so they get distributed nice and evenly. So there's photosynthesis happening all over the cell nicely like that. I'm breaking up. Oh, I'm sorry. Can anybody hear me?
Yeah.
Yeah.
Oh, the formula was a reduced version. I didn't put the numbers in. It's 6 CO2, it's 6 water, makes 1 glucose and 6 oxygen. And it's 6, you know, it's 6 is in respiration 2. Somebody just called me fine. Wow, I'm going to tell my wife that happened. She'll be all like jealous. People are calling me fine now. Honey, you should hear what my students are saying about me. Yeah, now she puts, never mind. Oh, thanks. Thanks. So much for my self-esteem. Anyway, what am I on about here? Oh, yeah, chloroplasts. So in the chloroplasts, then we have these thylakoid discs here. You know, we have these little discs, they're thylakoids, and that's where photosynthesis takes place. Stacks of them are called grana, or one is a granum. And a thylakoid membrane is where the light reactions of photosynthesis happen. That's really where the light gets absorbed into chlorophyll, and that's where, you know, the beginnings of the 2 steps of photosynthesis happen. I don't want to say the more important part. It's that both parts are equally important. Like this is the beginning where you make energy molecules like ATP and NADPH. And then the second part, which is the dark reactions or the Calvin cycle, it really is what we should call it, happens in the stroma. So the stroma is like all the stuff between the thylakoids. That's where that takes place. And that's where then the CO2 is absorbed and made into sugar. And the energy to do that comes from the ATP and the NADPH from the light reactions. But all of that stuff happens on these membranes right here, and we're going to go look at that in much more detail in just a second. So that's just how it would look on an actual electron micrograph, okay? No big deal. But this picture is really important. Like this is nice and easy to understand. It's super simple. It's big picture. This is like what you would need to know for the College Board for the test. This is the one that you really want to focus on when you're reviewing, you know, before May for that test. For me, you have to know more. I'm sorry, but you know, I know I'm making it stressful, but I can't help it. I don't want to make this class too easy. You know, I don't want people to say, oh yeah, it's a blow off, you know? So anyway, light reactions in the thylakoids absorb light. So light's coming in here. Whee! Oh, I should make it green because it can't be green light. You guys know why. I'm going to make it black because that like implies all the colors, even though white light is all the colors. Okay, so light comes in. It causes some stuff to happen in the membrane. That membrane, then those enzymes on the membrane will produce NADPH, which is really similar to NADH. It's a high energy molecule, and it'll make ATP. Okay, now the function of NADPH is a little different than it is for NADH, because NADH was like the thing that we use to drive protons. We're not using it for that anymore. We're using it to build glucose. We're using it to build sugar. So the function of that is different. That's going to happen in the Calvin cycle. The other thing we need to make the sugar is ATP. So those two are necessary. The Calvin cycle needs those. And obviously the Calvin cycle needs CO2 because that's the carbon, you know, source to make the glucose. Technically, the Calvin cycle doesn't make glucose. It makes glyceraldehyde 3-phosphate, but that's like half a glucose. 2 of those make a glucose. So it's like a horse apiece. Who cares? NADP plus, which is low energy. then goes back from the Calvin cycle to the thylakoids. ADP, which is low energy, then goes back to the thylakoids and gets recharged. So on the thylakoid membrane, the light reactions are going to recharge this into this, and they're going to recharge ADP back into ATP. And the way they do that with the ATP is just like the mitochondria do with ATP synthase. It's just like mitochondria. ATP synthase is powered by proton gradient. It's chemiosmosis again, just like in mitochondria. It's very, very similar. I told you there's some similarities. All right. Okay, sometimes people call this the dark reactions. Got to be careful with that because that's really kind of misleading because the dark reactions do not happen at night. I hope you guys understand that in order for the Calvin cycle to happen, it has to have ATP and NADPH. It has to have that. And at night, when this is shut down, this is also shut down. So the Calvin cycle cannot function at night. It's off at night. So there is no dark reactions. They also happen in the light. They just don't need light directly. They just need those two energy molecules. So be really careful into thinking like, I've heard teachers, by the way, I've heard teachers say that the dark reactions happen at night and that the light react. That's not true. I'm not one of those teachers. I tell you the truth most of the time. All right. Everybody good so far? By the way, in the light reactions is where we make oxygen too. Oxygen is actually a waste product here. It's garbage. The plants throw it out. Well, it's a good thing for us because we love oxygen. We're kind of addicted to oxygen. So anyway, gotta have it. Any questions on this? All right. That either means that you're too depressed, can't talk. You're too depressed to ask questions, or you understand it really well and you want to move on. It's one or the other. Hopefully it's the latter. All right, let's talk about light a little bit. I don't want to get into like huge physics discussions here. Like that's Carruthers' turf, you know, and Mr. Shane and those people teach physics. I do think it might be a nice idea to talk a little bit about the nature and the physics of light to understand, you know, photosynthesis a little bit better. I need you to understand that white light, like sunlight, is actually all the colors of light. It's all the colors of light mixed together. So white light is not really white light. It's all the colors of the rainbow mixed together. Okay, the old Roy G. Biv, mix them all, you get white. As a matter of, please don't leave. As a matter of fact, you only really need 3 colors to make all the colors of light. And those are the 3 primary light colors. Do you guys know what those are? Everybody knows this now because everybody has these like things on their Christmas tree and in their room and you know on the ceiling and on their cars. And anybody know what in their computer case, on their skateboard? You guys know what I'm talking about? Somebody said red, blue, and I forgot. Yeah, it's red, blue, and green. So it's RGB. Have you heard of RGB? People always say like, hey, are you gonna get some RGB for your Christmas? Nobody ever says that, but like people in the car world are putting like RGB lights on their cars, so it lights up real cool. You can make any color you want with the RGB because red, green, and blue mixed in the right way can make any color, which is pretty awesome. Like you might have bought one of those LED strips that you can stick on the wall, and then you can have the little remote, and you can make all the colors with the remote. And that's how that works. Because there's also your TV or computer monitor, your Chromebook monitor only has. It's not yellow. It's yellow is a pigment for paint or for ink. So we got yellow, magenta, and cyan are the 3 primary pigments. That's different from the 3 primary colors of light. Pigments subtract light. Light adds light. So it's that I don't want to get into in huge detail, but that's actually kind of interesting but also a little bit confusing. We're not going to worry about that too much. So I need you to understand that chlorophyll, no, you're not going crazy. Chlorophyll is really, really good at absorbing blue and red. Chlorophyll in chloroplast is really, really good at absorbing red. And if it absorbs blue and red, the thing that's left is green. So that explains why plants are green, because plants do not absorb green light. So there isn't anything like inherently green. It's just that the chemicals like the chlorophyll pigments and all the other pigments that are in there they take all the blue away, they take all the red away, and the only thing that's left is green, and that reflects because they weren't able to use the green. Now there's evolutionary reasons for that we don't need to get into right now. It has to do with, okay, I'm not going to get into it, but if you take away the blue, if you take away the red, all that's left is green, and that reflects, so it looks green. So any kind of object that you have, if you're looking at, like this face mask that I have, it's blue. The reason it's blue is because it's absorbing red and it's absorbing green. All the red and the green is getting absorbed by the paint pigment or the pigment, whatever they use, dye pigments in this cotton, and then the only thing they let go is blue, and then the blue gets reflected, and that's what we see. So that's how that works. So I need you to understand. Yeah, more like chlorophyll. That's true. Wait, you can't see this? You see this or not? Yes? We can see it. Okay, thank you. Everybody clear on the color business and the pigment business? So let's take a look at chlorophyll. Let's take a look at chlorophyll. So if you look at the absorption spectrum of chlorophyll, it's basically, we're just going to look at chlorophyll A and B. Forget about the carotenoids. The carotenoids are more like the orange or whatever color, like in carrots or whatever. Chlorophyll A and B have a big peak at blue and another big peak at red. Okay, so it's basically, if you look at the spectrum, blue is absorbed really well, red is absorbed pretty well, and then green is not absorbed very much at all, very, very little, which means if you shine green light on your plants, they're not gonna work. They're not gonna do photosynthesis, they're gonna die. Like if all you give the plants are green light, it's like giving them nothing. It's like giving a person just water as food. Like water is useless to us as food, there's no energy in it. And green light that can't supply any energy. It has energy, but the plants just can't use that wavelength of energy. They can't, chlorophyll doesn't work for that. It doesn't get activated. All right, so if people are emailing me right now, I'm not looking at my email because I keep hearing bing, bing, bing on the email. So I don't know, do I need to look at that? No, I don't want to break out of this. Action spectrum is like actual, so this is absorption, that's the absorption spectrum. This is actually like how much photosynthesis is happening by measuring oxygen getting released. So blue light, lots of oxygen. In other words, lots of photosynthesis. Green light, very little photosynthesis. Red light, lots of photosynthesis. So that's my action spectrum, actually measuring photosynthesis. This is measuring actual light absorption, but they're parallel. They kind of match each other. This guy, Engelmann, he did an experiment where he so he took white light and he broke it up with a spectrum into all the colors of the rainbow. So Roy G. Biv all along the way. He then shined that on this filament of algae, which did photosynthesis very similar to the way plants do it. And then he put aerobic bacteria, which would only grow if there's oxygen getting produced. And in the blue light algae parts, the bacteria grew really well. In the red light algae parts, the bacteria grew really well because plenty of oxygen was produced here and here. And then in the green light, very little bacterial growth because they needed oxygen in order to grow. So that kind of demonstrated that the absorption spectrum actually reflects what's really happening with photosynthesis, and it directly relates to photosynthesis, and that it all makes sense. Well, when you're this popular, a lot of people, want to be your friend, so it's rough. Well, we can look at this. Let me just see here. Why don't I go into the light reactions right away? I'm going to draw you a picture, which hopefully will turn out all right. Maybe we'll see. And in the picture, there's going to be a complex like this. On the outer area, the light harvesting complex, I don't need you to know those terms. There's pigment molecules like chlorophylls and other pigments too. They're all over the place. We have them colored green because they would look green to us because they absorb red and they absorb blue and they leave the green behind and reflect the green. So that's why they look green. So red light and blue light will get absorbed and activate these. And they get so active that they then What happens is an electron jumps up one energy level, which I know you guys all learned in chemistry. It jumps up one energy level, and then when it drops back down to that same energy level, it releases another photon. So a photon comes in, excites the electrons in this thing, which then release another photon, another photon. So there's little flashes of light flashing through this complex right here until they get to a very special pair of chlorophyll molecules. And in the first photosystem we're going to talk about, which stupidly is named photosystem 2, that is called P680. Because it absorbs at 680 nanometers really well. Hold on a second. Hello. Yep. Okay, I'll email it to you. All right, bye. Got to take a time out. So why don't you just look through the slides a little bit, and I'll be back in like 2 minutes. Thanks for being patient. Okay, so there's a special set of chlorophyll molecules in the first photosystem, which is called photosystem 2, stupidly, that are called P680. That's not really all that important. I just need you to, I'm just going to say it anyway, because there's another set of them in the other photosystem that are called P700, and they're slightly different, but they, kind of similar. And that just has to do with the wavelength of light, the color that they absorb best, but that's not even anything that we need to worry about. What happens in the first one is that P680 is actually going to take water and split it. It's going to split the hydrogens and the oxygens of water apart. So it's going to take water and split it into oxygen gas and hydrogen ions. And it's going to take the electrons that are now missing from that process, and it's going to shoot those electrons up into an electron transport chain. You're like, what? Electron transport chain, just like mitochondria? Well, kind of similar actually, with similar components, but different enough, to point out. This primary electron acceptor is just really an electronegative. It kind of sucks the electrons up this way and then feeds them through into another molecule. So what we're going to do is take a look at the big process and look at all of it, and we're going to try to draw it, okay? Now, you can always look at pictures. I have pictures on my Google Drive. I have pictures for you in that resources thing. I still would like you to draw it out on paper, though, because you learn it better that way, and you're going to remember it better. This shows the energy relationships here. We may go back to this later, but this is pretty much just review. I'll go over this again later. This is like the fancy book picture, so we're not going to use that right now. What we're going to do is use my homemade picture, okay? So, and you, okay, do you want me to draw it from scratch? Or I kind of want to use this. I like this. When you draw it from scratch, you don't have to make it this complicated. You could do this part just like that. Draw these two guys right here like this. And then draw this thing up here, and that's all you need. And then you can put your chlorophylls here. And that's all you need to do, so that's easy. The rest of this is pretty easy to draw. So I'm actually going to be lazy and use my pre-made paint picture. And I can do that because I made it. So it's not like I'm stealing from somebody else. I made this thing like, I don't know, 20 years ago in Microsoft Paint. And that's why it looks so stupid. All right, let's start in the beginning. First of all, we're in the thylakoid membrane. Okay, so if we're looking at a chloroplast, I'm going to erase this again, and the thylakoids are right here. PowerPoint really hates me. We're talking about this membrane right here, which means this up here is the stroma. We're in the chloroplast in the stroma, and down here is the inside of the thylakoid. All right, let's see if we can get her in here. So this is the inside of the thylakoid. Now, or the inner thylakoid space, I labeled it here. Okay, and here I labeled the stroma. All right, cool. So now we know where we are, and now we're going to see what happens. She get in? Nope, she did not get in, so that didn't work either. Okay. All right, we're going to start with a photon of light. So one particle of light, and the nature of a photon we don't need to worry about, like the whole nature of particle, wave matter duality, whatever. Let the physicists worry about that. We're going to just say one beam of light. How about that? One beam of light, which is like one photon, one particle, is going to hit this chlorophyll. The light beam is going to get bounced around like I described to you. It's going to get fed into P680. This whole thing is called photosystem 2. The reason it's photosystem 2 is because photosystem 1 is more ancient. It evolved first, and I think it might have been discovered first. So when it was named, it's named backwards now. Like, the first thing that happens is photosystem 2. It's kind of goofy, but it's, now that I told you how weird it is and how dumb that is, but it's easier to remember, hopefully. Oh, she made it in. Nice. Hey, Claire, did you make it in? I think. I hope I heard a ding ding. Maybe not. Oh yeah, there she is. Awesome. Okay, so that code worked. Thank you. All right, so then what's going to happen is the P680 has a special talent that very few molecules in nature have. It can take water and split it. So what's going to happen is water is going to literally get split up into 1/2 O2 and 2 hydrogens. Okay, and the 2 electrons from this, they get fed into this system and they get sort of flung through this chain now. Okay, the primary electron acceptor sort of attracts them and they go this way and then they fly through over here. That's going to disappear again, isn't it? Oh, it worked. Into a molecule called PQ, which is plastoquinone. And they're all described for you down here. Or they're all in your book too, but those are right there, plastoquinone. So, oh, that did go away. Okay, so then it goes through, nope, that's bad. Hold on. All right, it goes through here into plastoquinone and it feeds into cytochrome complex all the way into plastocyanin. And from there, it goes this way. Okay, so what happens is as these electrons flow through these different molecules, through these proteins here, what's going to end up happening is they power hydrogen ions to get pumped into the thylakoid. So hydrogen ions get pumped into the thylakoid, just like in the electron transport chain of mitochondria, where hydrogen ions got pumped into the intermembrane space. So it's like, hey, that's kind of similar. We already know this process. That's kind of cool. It's not one hydrogen ion that gets pumped. It's a lot of hydrogen ions. Don't think it's just one, lots of them, because they only get pumped in one spot here, whereas in mitochondria, they get pumped in 3 places. Now there's only one, but it's a very powerful pump that pumps many of them in here. And I think it's 4 per 2 electrons or whatever it might be. I don't really care. We're not going to do a ATP count for this like we did with respiration. Okay. So plastoquinone, we got cytochrome complex, which is reminiscent of cytochrome C in the other electron transport chain from mitochondria. Slightly different, but somewhat similar also. And then by the time they have pumped this hydrogen, these electrons are spent. They're low energy now. They don't have a lot of momentum left. They can't do a lot of work anymore or any work. So we have to charge them up again. So as they enter this second photosystem here, what's going to happen is, try that one more time, see if I can do a better job here. They now enter this photosystem 1 over here, and they get charged up again by another photon of light. So this photon is bouncing around over here, very, very similar to this other photosystem, and now it charges the electrons up again. But this time we do not split water. This time the energy of that photon is used for something else. Now these electrons once again get thrown or thrust through this primary electron acceptor and then into, oopsie, sorry, I'm back. And then into a molecule called ferredoxin. And from ferredoxin, they go through this NR molecule, which I'll tell you about in a minute. And then they leave the membrane and they get brought out here where they get accepted by NADP+. So NADP+ is the electron acceptor here, plus those 2 electrons, plus 2 hydrogen ions. No, 1 hydrogen ion, sorry, that's a mistake. Makes NADPH. Okay, so that's the purpose of the electrons. That's why they need to be that high energy. That's why they get thrust through here into this basically NADP plus low energy molecule to make NADPH, which is high energy. Has a lot of energy, and now that is going to be available for the Calvin cycle to do the to make the glucose building, or the sugar building, I should say. All right, so we got photosystem 2, we got plastoquinone, we got cytochrome complex, we got plastocyanin, we have photosystem 1, we have ferredoxin. All of these are listed for you here. And then we have, oh, this one I didn't say yet, that's NADP plus reductase. And NADP plus reductase just simply means it's reductasing NADP plus, means it's giving it electrons. It's giving it electrons, and by giving it those high energy electrons, it charges it up and makes it high energy, and it grabs a hydrogen in the process to stick on there to make that high energy compound. So this is high energy, that's one of the products, and then the light reactions have a second high energy product that we're going to see right now. So you might remember that we've been pumping hydrogen ions. Here are the hydrogen ions. Oops, lost my mouse. Here it goes. The hydrogen ion concentration is very high in this space now. So those hydrogen ions, they pass through ATP synthase, they cause it to spin, and that spinning motion energizes ATP synthase. I swear it's when I click this. That energizes ATP synthase to then produce an ATP, another high energy molecule, is made by ATP synthase right here. Obviously, we need a phosphate also. So inorganic phosphate is floating around, and that gets stuck onto the ADP to make ATP. All right, so those are the 2 main products of the light reactions, NADPH and ATP. That's the thing we make. The thing we have to put into the light reactions are NADP+. We have to put in ADP. Those all have to go in order to do this. And we have to put in water. None of these are high energy. Water is low energy. NADP+ is low energy. ADP is low energy. Where the heck are we getting the energy to make these high energy compounds? Oh, you know it. Say it. The energy comes from Does this help?
The light.
The light. It comes from the sun. It comes from the sun. Okay, so that's driving this process. Here, photosystem 2. Here, photosystem 1. Beautiful. We love it. Okay, normally, if the world was a normal place, I would say memorize this. Well, the world is a weird place, and everything is weird, and everybody's weird in the head right now, including me. I'm still going to say memorize it, but keep in mind, if I give you a test on it, you could theoretically have your notes available. But I kind of still want you to have it memorized. So I think that's helpful. It stresses your brain, and it makes your brain, you know how like when you exercise and you lift weights, it stresses your muscles and it causes like little muscle damage that then makes your muscles stronger and bigger. That's how you build up muscle. Well, by stressing your brain and doing something that's unpleasant and hard and takes a lot of effort, I'm not saying that's going to cause brain damage, but it's going to make your brain smarter. So I'll give you granola. Do I look like a granola type of guy? Really? Do you count the photons as something you would put into the light reactions? It's not a material. So the photons are not matter. They're not molecules, but they're an energy source. They are a wave. Okay, so in terms of like, is it a material that gets put in? No, but it is something that has to go in to make this work. So do you count the photons as something you would put in the light reactions? I would, sure, but just keep in mind that it's not matter. It's not a molecule. The bell keeps happening because my internet sucks. Oh, okay, got you. So the muscle damage in weightlifting is damage to our mental health. Sure, right. So your mental health degrades as you're memorizing photosynthesis, but your smartness goes up. Actually, there's like research that says that the smarter people are, the more like depressed they are a lot of times, because they understand what's going on in the world, and they understand the big picture of like, you know, repercussions of certain things and all of that stuff. So like one of the curses of being smart is a lot of times that comes along with being more depressed, which that kind of sucks. I mean, but you know, time to go dumb. That actually is true. Yes, lower IQ people tend to be less, they happy-go-lucky. Nothing phases them. Yeah. Yes, you're depressed because you're so smart. That's the silver lining. That's the good news. Yeah. Anyway, okay. Dude, we got to talk about Kelvin cycle here. Actually, let me, we got a little bit of time left, so let me see if there's something else on those previous slides I want to talk about. This just is a fancy picture for that. You might say like, oh, you know, ATP synthase is wrong because it's facing down, and on yours it faces up. Well, no, they just had the membrane curve around, so it's facing out of the thylakoid. That's what matters, out of the thylakoid. Okay, so if I flip this around, I mean, that would be facing up into the stroma. You know, stroma is here and stroma is here. So, I mean, that's not really an issue. And then, you know, what's awesome is, look at this, NADPH is made here, ATP is made here. The friggin' Kelvin cycle is like right here. It's like right next to it. So the ATP is made right where the Kelvin cycle needs it. So that's awesome. That's awesome. Am I allowed to say friggin'? I don't know. Well, okay. What else did I skip? Oh, these guys. Basically, this just shows the energy of the electrons. So the electrons come in at high energy because light gives them high energy. So high energy higher up on the tower is high energy. Now they flow down this ramp and they lose some of their energy because they're making ATP synthase. So now the electrons are lower energy. They got to be charged up again. So here comes photon guy number 2, pounds down on this, you know, teeter-totter and jacks them up again. So now they're high energy again. Now that high energy goes into making NADPH. So you get this nice high energy NADPH compound at the end. I like this picture. It's kind of nice. It's confusing at first. This just shows the same thing about how all these processes and how the energy flow works. So you can look at this if you'd like to, but it's basically a simplified version of what we have here. All right, cyclic electron flow, let's talk about, not right now, or do we? Let's talk about it later. I'll probably forget to talk about it, whatever. It's like a thing that some plants do when there's bad conditions and when there's not enough water and stuff like that. It's like an emergency sort of approach, and real primitive photosynthetic organisms did this. They didn't have photosystem 2, they only had photosystem 1. And they made ATP with just photosystem 1 by having the electrons sort of go around in the circle instead of ending up in NADPH. So it's a more primitive, simple way of doing photosynthesis that some plants can still do now, or maybe a lot of plants can still do this. But anyway, yeah, I might not ever mention this again. But yeah, Kelvin cycle. That's something worth talking about. One of my former students from many years ago got actually hacked into my PowerPoint and drew Kelvin's bicycle. His name was Joseph, or I called him Broseph, but he made it into like an old-fashioned bicycle, which I thought was kind of funny. So I kept it in here. That slide's been in here now for 7 years or whatever. So, but he must have been on my computer. He was my TA, and I must have been going to the bathroom or whatever. And he went on my computer and he changed my PowerPoint to this. So I saved it. This we're going to learn how to draw. So why don't we do that now? I got to insert a slide. Sorry, I thought I was ready for this. Let's do a slide. So Kelvin cycle, pretty interesting stuff. And it relates to the light reactions so nicely. It just all makes so much sense. It's great. I love it. Sometimes I got to teach you stuff that's kind of confusing and it hardly makes sense to me, but this stuff it's very intuitive. So this is nice. Let's go with black. All right. So CO2 feeds in. CO2 obviously comes in from the air. There's CO2 in the air. There's a certain amount of, small percentage of CO2 in the air all the time. It comes from various sources. It comes from combustion. It comes from respiration of animals and, organisms. So that CO2 comes in through the stomata and it reaches those mesophyll cells and it diffuses into the cell. And eventually it makes its way into the stroma or the chloroplast. So it gets fed into the Kelvin cycle by an enzyme that we call rubisco. Rubisco is able to grab the CO2 and incorporate it into organic molecules. As a matter of fact, what rubisco does is it takes 3 molecules of RuBP, which I know you want to know what it stands for. It stands for ribulose bisphosphate. RuBP is kind of nice. Calling it RuBP sounds nice. Kenny Rogers loved photosynthesis so much. He's a country singer. He wrote a song about RuBP. He wrote the song RuBP, Don't Take Your Love to Town. Ask your parents. They'll know that. You guys are too young to know that, old guy. All right, so 3 RuBPs with the help of this enzyme rubisco, which stands for ribulose bisphosphate carboxylase, grabs the 3 CO2, oh, no, hold on a second. 3 CO2 molecules, and it makes 6 3-phosphoglycerates. All right. So right now, let's just do abbreviations. We can write them all out later. So the 3 RuBPs and 3 CO2s makes 6 times 3-phosphoglycerates. Each of these is 3 carbons, which gives me 18 carbons total here, yes? There's 3 times 6 carbons, or 6 times 3 carbons makes 18 carbons. Well, this is only 3 carbons. So if I take 18 minus 3, it tells me how many carbons are over here total, right? 18 minus 3 is 15 carbons. That tells me how many carbons on our RuBP, because if this is 15 total, and there's 3 of them, how many carbons is RuBP? Shout it out. 5, yeah. Each of these is 5 carbons. All right. So 3 5-carbon molecules makes 15 carbons, plus 3 carbons from CO2, because there's 3 CO2s, makes 6 3-phosphoglycerates, 18 carbons. So this is a kind of a simple 3-carbon sugar that is not quite ready to make glucose. We got to convert it. We got to make it. We got to enhance it a little bit. Okay, so what we're going to do is take the 3-phosphoglycerates and we're going to make them into 6,1,3 bisphosphoglycerate. And the way we do that is obviously with ATP, because if we're going from 1 phosphate on each of these, so on carbon 3, there's a phosphate. There's only 1 phosphate on each of these, and there's 6 of these. And now here we're going to have the same thing, but now there's 2 phosphates on them. That means we need 6 more phosphates. So the way we get the 6 phosphates is how? We need 6 ATP. Where in the world are we going to get 6 ATP from? Well, I know we get it from the
Right.
light reactions, or some people call them the light dependent reactions. Okay, we get it from there. So we got all kinds of ATP that was made over there. We're going to use it up here. I'm going to get rid of this 18 carbon thing. That's super annoying there. All right. So those 6 ATP then get converted to 6 ADP. And I don't put the phosphates, that's supposed to be a D. The phosphates don't come off over here because they're now stuck to these. They're stuck over here. So the 6 phosphates from the ATP are over here now on these molecules because there's now 2 on one on carbon 1, one on carbon 3. That gets converted to 6 glyceraldehyde 3 phosphates. And the way we do that is we need energy again. We do it with NADPH. So 6 NADPHs come in and form 6 NADP pluses. Okay, so that's how that works. Where does the NADPH come from? We already know the answer. Why? It comes from
Light reactions.
light reactions. Yes, thank you. I saw your note. I saw that. Thank you. I'm being a hypocrite, not listening to you guys. 6 of these go to 5 glyceraldehyde 3 phosphates, which then get converted with the help of 3 ATP into 3 RuBPs. So 5 glyceraldehyde 3 phosphates, that's 15 carbons because it's 5 times 3 carbons, makes the 15 carbons of 3 5-carbon RuBPs. All right, so that's 1 glyceraldehyde 3 phosphate left over, 1 G3P is left over. That leftover G3P is what we make glucose out of. That's now donated to the cell to make carbohydrates, to make glucose, to make amino acids, to make fat molecules. That's your sugar that we make from photosynthesis. So photosynthesis technically does not make glucose, it makes G3P. But if you do the Calvin cycle twice, you get 2 3-GPs, and you can make 1 glucose out of that. So if you do Calvin cycle 2 times, you get a glucose. If you do it once, you just get a G3P. Everybody clear on that? Now, normally, in normal times, I'd ask you to memorize this. This is weird times, still going to ask you to memorize it. So please do. I memorized it, and I'm old and decrepit, which if I can do it, you can do it too. By the way, all right, so let's take a look at the lab I want you to do. I have a little bit of time to explain it. Hopefully it'll be enough time. It's not letting me escape. Thank you, PowerPoint. Apparently you hate me. Let us take a look at Google Classroom. I think did I post the lab already? Yes, I did. So I don't have this done yet. I started typing up the write-up, guidelines, and I didn't get done. I spent 3 hours on this and I'm still not done. So what took me so long is not something that's going to take you that long. It's going to take you way less long because I had to make the Google sheet with all the calculations and the graphs. I configured that all for you, and that took me forever. And I went through the whole lab. So I did everything that you did. Like I did all the reactions, all the measurements, I did everything. So that didn't take super long, making the Google sheet did. And so I'm not done with the instructions yet. I'll post those hopefully by Monday. I'll post those by Tuesday maybe. And we'll talk about when this is due. I think because we have another lab, a photosynthesis lab coming up, I think I'm going to have to make some of these labs do like after the test. If that's okay. Maybe even like over winter break, I could have you work on them. So you're not like too freaked out about having to get 2 labs done before the test. We'll be reasonable about due dates on those. Yeah, okay, all right. Let's click on, so this is like the data entry sheet. Okay, so you'll need this to collect your data. So we're gonna be working with crickets, a mouse, and garden peas that are germinating. And it's all set up already. All you got to do is enter your data. So you just make a copy of this for yourself. I think my Google Classroom made a copy for you. So then what you do is you go to Pivot Interactives, log in, please. And hopefully everybody knows their login and everybody hopefully has the right email in there. So go to the Exploring Respirations Rate lab. That looks like this. Okay, so if you look at my labs here, it's this one. It has a very flattering picture of me. That's the one you click on. Okay, and I chopped off all the extra garbage. This had like tons of pages of questions and like intro and all this stuff. I cut it all away. It's all gone. All I have is this left where you have to do the the video measuring like you did before. Okay. I don't remember who made that picture. One of you guys made that. It was like the class from last year, I think, made that. Noel did. Oh yeah. Okay. All right. So this is what we're going to do. So we're going to be measuring respiration rates of crickets first. So you can adjust that here. You can load either a mouse, crickets, peas, or a rat. We're not going to do the rat. We'll just skip that just for, it's kind of redundant with the mouse. It's just a bigger mouse, so it's stupid. So we're going to skip it. We're just going to do crickets, mouse, peas. So it's already at crickets. You can select the temperature here, cold, normal, warm. Okay. So it's either, and we're just going to say it's 12 and it's 24 and it's 31. They always do that range, which is super annoying. So we're just going to say 12, 24, 31, done. So we set it to 12 and we load in, always click load. So it comes and it's already in. Okay. So now let's make this go away. Cancel. Okay. All right. So now we're ready to run it. We got to take our measurement of parts per million CO2 at 0 time. So this is 0 time 582. So I would now go to my respiration rate. At 0 for the crickets, I would put in, what was it? 582, did I say? I would put in the 582. Yep, so that's what I'm going to do. So I'm going to delete that again because I don't want you to get my data. Wait, I don't see it. You don't see the lab? Oh, I didn't post it. Oh, I'm an idiot. Okay, let me go back. I'll post it. Sorry. I'm sorry. So sorry. I'm sorry. You guys could shout into the mic, so I don't have to read the chat to see this, because now I wasted 3 minutes. You could say it, so I hear you. Does it show up now? Refresh or something? My hand is numb.
Yeah, it shows up now.
Leilani, I think we had a lot more fun in Honors Bio than they did this year. Trust me. Our Honors Bio last year was 10 times more fun than what they're getting. So the fact that they got to play Kahoot one time, don't worry about. Anyway, I don't know if they get any of my jokes. Like I don't know. We can play Kahoot in AP Bio if you want sometime. I'll do that. That's pretty fun. It's like Jackbox. So we can try that sometime. All right. Okay, everybody in or not? You gave me no feedback. Everybody's, okay, so you go to the video thing here and you feed in the right info and then you start recording. So 582, you go to the thing here and you type that in. Okay, then you go to, you just fast forward. You go right to 5 minutes here. Hit play, go right to 5. Oh, you got to set the clock up. You got to put the clock in here. So here's my clock. I go to 5 minutes, as close as you can, 5.02, perfectly fine. Now it's 7:07. I go to my thing. I type in 7:07. Okay, so this data table down below is always going to subtract the initial ppm from the next one. So it's going to just measure for you the change. That means for the first reading, it's going to read 0. That means at 0 time, 0 extra ppm has been produced, or extra CO2 has been produced. So this table down here, you don't have to do anything with it. I already set it up for you that it's going to subtract automatically, and it's just going to show you the extra CO2 that was produced since we started. Okay, then you go over here and you switch it to, let's say, mouse, or no, then you go and you switch it to the normal temperature. You load and you do it again, all the way up to 20. Then you do it again. You load the warm temperature, 31, and you go up to 20 and you take your measurements, 0 through 20, every 5 minutes, just like what the table tells you here. Now for the mouse one, it's a little weird. They only go up to 15 minutes. So you can only collect data up to 15 minutes. Well, that's what you put in then, or 10 minutes maybe. I think it only goes up to 10. So then you only put in the 10. Okay, you do that for the mouse. Notice here, my number zeros here, enter here, right here, because you can only enter this much data right here. And then for the P's, there's something funny too, where it doesn't go all the way to 20 on one of them. Just enter as much as you can. You'll see how that goes. And that's as far as we are right now. And then you can start thinking about that a little bit, like why did these results turn out this way? Why is it different for the mouse and the P's? And then the other thing I want you to think about is, For the peas, we put in like, if you take a look here, 264 grams of peas. That's like half a pound of peas. The mouse only weighed 16 grams. That's half an ounce. Is it really fair to compare half a pound of peas with a half-ounce mouse in terms of respiration, CO2 production? So we really need to figure out the rate, which is the slope of the line that's going to form, per gram of organism. So we do that calculation, and I did that automatically for you also. That's already done down here for you, okay, in this table. I just need you to understand why I did that. So you don't have to do the calculation. I did it for you. It's automatic. But I need you to understand, why do we need to worry about per gram? Okay, so we're at 1:02. We're 2 minutes after. I'd like you by Tuesday to have all this data collected. So do that this weekend. Get all the data collected for all three animals, all three temperatures. That's going to take you about 30 minutes, maybe 20 minutes or whatever. I don't know, maybe half an hour. It's not that big a deal. It's easy. And then you don't have to do any write-up yet, but I want you to start thinking about the lab, like, why are these results so weird in some cases? Like, why is this not what I expected? And why do we need the per gram respiration rate? Why is that important? Especially looking at stuff like crickets, 48 grams. That's way more than the mouse. Peas, half a pound. That's a huge amount. So then you need to think about, why do you need to divide the mass? Okay, that's all we're going to do for class. Several of you owe me remediation. You should be coming into TSS. I have a test prepared for you for remediation. You know who you are. I expect to see you in TSS. If you're not showing up for TSS, I'm assuming you're happy with your grade and you're just going to keep it where it is. Okay. A bunch of you still owe me video evidence of your test analysis meeting with each other, where you Zoomed each other and you talked about the test and you recorded that 5 minutes, and you're going to post that to me. So I see that. So make sure you get that in, and have a good weekend. Have fun with the lab, study for the test, and we'll finish photosynthesis on Tuesday. Bye. Tell me what to do, Buckner. Have a good weekend. How dare you?
Where do we post those videos?
Huh?
Where do we put those videos?
You can just,
Because there's no spot for them.
I know. You can just simply, I'll tell you what, I'll make a spot in Google Classroom. I was going to say, just share it with me and email. I'll make a spot.
I tried emailing it to you, but it's too big.
Oh, I'm sorry. Okay. No, don't email it to me. Just share it with me.
Okay.
Don't actually send me the whole file.
I can try to.
Yeah. Okay. I'll put it in Google Classroom. Okay.
Okay.
Then it'll be like an assignment that you can do. All right. Thank you.
Bye.